Hello lovers, in this video Dr. Edwards is going to be going through genome sequencing, the long story of this, the process of how we decided to do this, how it happens, everything that was involved in this. And this is for your A-level biology. Now this is an absolutely fascinating process, and if you want to make sure you've got the knowledge in your head for your exam, then over on the website, once you finish watching this video, you can do the set of questions that Dr. Edwards has written especially for you to make sure you understand everything thoroughly.
DNA sequencing. So the first ever method for sequencing could only do fragments of DNA up to 900 base pairs. The fragments were then aligned based on portions that overlap to assemble large pieces of DNA. And this method was used to sequence the human genome. Bear in mind, we ended up sequencing millions and millions of base pairs, not just only doing it 900 at a time. So this was very long and labour intensive. You had to split the DNA into 4, and then each portion replicated with a different radioactive nucleotide, so the fragments could be detected and then separated by gel electrophoresis, and then an X-ray taken of the gel by hand to view the fragments. So this was very, very long, very slow process. And over time, faster sequencing methods now exist in more recent time, and they're continuing developing to become more efficient, more powerful, and cost-effective. So the old methods were labour intensive, they were very, very expensive, and they could only be done on a small scale. Whereas now we have automated machines and computers which can work much faster than humans can. They're cheaper, so whole human genome sequencing is now only costing around £1,000. It may even be less than that. And you can send away one of those test swab kits to get some of your DNA sequenced and then or checked for various genes for ancestry reasons or health reasons. That costs you about £100. They're more cost effective to do large scale, so we do things on a large scale now rather than doing them on a small scale, and that means doing large scale projects like the Human Genome Project can obviously be done much easier and faster. All of this combined together has made what we call high throughput sequencing possible. So we're doing things thousands of times faster, thousands of bases faster, and for a fraction of the cost, which means that sequencing genomes is now really easy to do. And we've got various different methods to use and lots of options, but all of it is now automated and easy to kind of make sure that we can get this speedy sequencing done. So this is an example of one of the more up-to-date cycle sequencing methods that was used. It was developed after the Sanger method, which was that method we were talking about used for the Human Genome Project, and it was just kind of updated and made slightly better and faster. So in our reaction mixture, we have primers that are able to bind to the DNA. And then we have our template DNA, which is obviously what we're trying to sequence. And then we have these special nucleotides. So the synthetic nucleotides have a fluorescent tag on them. So there's 4 different colours here. We've got orange, blue, green, and pink. And then we have DNA polymerase enzyme, which is going to add these nucleotides and also normal nucleotides, non-synthetic nucleotides, to the DNA strand after where the primer is bound to. So it adds nucleotides until it adds one of the fluorescent nucleotides. When it adds a fluorescent nucleotide, it stops. And so that fluorescent nucleotide becomes the end or the terminal nucleotide in the sequence. So you can see here I've got loads of different fragments, and at the end of each one is a different fluorescent tag. So that ends with a different base. This is done repeatedly, so we get all those fragments built up, and then we send those fragments through a very, very thin capillary tube, and it separates them by size. So the smallest ones will go first, and the largest ones will go last, because due to separation techniques, much like chromatography or gel electrophoresis, which we're going to look at. The laser they have to pass through reads or detects the colour of the fluorescence of the terminal end piece. So in the first of my shortest fragments, I've only got 1 fluorescent nucleotide. That's the first letter. So then the next letter where there's 1 nucleotide and then the fluorescent letter, that's the next letter of the base, and so on and so on and so on. So then we can then build up this sequence where we put all the fragments in the right order so we know what the sequence of bases are. And you can do that up to a certain base number size, but it's quite large, and so you can simple like this, and it produces this output really where you get the peaks of the chromatograph that we call it, but it's kind of really a readout of the peaks that the laser read of the colours of the spectrum, and then that determines which letter it is, and they'll tell you your sequence. And you can send samples away from the lab and they can sequence it, and they'll send you back the readout quite cheaply now, depending on how many samples you're doing or how long the base pairs are of the fragment that you're looking at. But this is a really easy way of kind of sequencing DNA. Okay, so that's quite a short one about genome projects and sequencing. You need to know what these projects are about, how they work. The sequencing, you don't need to go into so much detail, but you may get given a sequence to read, or you may get given the context around using sequencing in order to get or know or understand the base sequence of a section of a genome or the whole genome. Everything we're going to talk about in this kind of topic section about genetic technologies relies on us being able to understand the human genome or the genome of any organism that we're looking at, because we need to know the sequence of the DNA in order to be able to figure it out, understand it, know and find certain genes and what sequences they are, and then use that to help figure us code what they code for in terms of proteins. So we've looked at the genome before, and we should know that that's all of the genetic information in an organism or in a cell. So genome projects are just technology and projects that use technologies to determine the complete sequence of bases that make up the DNA of an organism. So it's about sequencing the entire genome of an organism, so we know all of the bases in the order that they exist in. The main example for this, and we will have probably talked about this at GCSE as well, is the Human Genome Project. So it was a collaborative effort of scientists all over the world. They sequenced 3.2 billion bases, which is obviously a lot. Previously, up until this point, the most that we managed to sequence in one go or in a chain was about 10,000. As a result, we now understand and know the sequence of the human genome. We can diagnose and treat diseases, and we would never have been able to do that without understanding and knowing these sequences. We can now try and trace causes of inherited diseases, and they can be found in days. So we can look for mutations in days instead of trying to understand it and figure out what mutations were causing that, which could have taken years beforehand. So this is a timeline. It took from 1990 all the way to 2003 to actually achieve this. But now genomes are sequenced in a couple of days. So genomes of other organisms, because of the technology advancements that happened during this time and the collaborative effort of everyone working on this technology, the technology has come forward so quickly and so fast, a sort of exponential increase in the abilities of the technology that we were using throughout this time, that now we can sequence things even faster. We need to know some of the applications that we can use these sequencing techniques for. So we can compare whole genomes of species now, using computers and techniques called bioinformatics, where it can analyse data on such a large scale. It can help us to actually track the differences between species, which can help us determine their evolutionary relationships and also determine how related they are to each other. As well as comparing different species, we can also compare human genomes to each other. This helps to identify mutations that can cause disorders. So by comparing the DNA sequence of someone that doesn't have a genetic disorder and someone that does, we can look for the difference and find the mutation that may cause the disorder. Once this has been identified and we know the sequence of the mutation, we can use it to screen people for that same disorder. It also allows us to study genotype-phenotype relationships. This allows us to maybe predict someone's phenotype based on their genotype. This could be, for example, symptoms. So by checking what type of mutation someone has, then we can use it, compare it to other people with the same mutation that we know what their symptoms are of their disease, and then we can predict what symptoms this person will have. This uses bioinformatic analysis. It's also the way that we can compare people's genomes to look for certain genes that give you certain traits when you send off for those DNA sequencing kits. We can use genome sequences to predict amino acid sequences. It relies on us knowing the knowledge of locations of introns and exons, however, and this isn't always possible and hasn't been fully done yet for various genomes. Another use for DNA sequencing is in the field of synthetic biology. This is where we're going to be using DNA genome analysis to provide a starting point to help us start to build synthetic devices and systems based around biological processes and biological systems. So, for example, we can use it to help encode information. So storing information on synthetic DNA. It's obviously a really great molecule that stores a code, and obviously we can use binary code and things like that to be able to store information and get a computer system to translate that. So it's a way of storing information on a really, really tiny molecule, basically, which is one way we can use this kind of knowledge of biological systems and the DNA analysis to actually produce something kind of synthetic that can help us in the technology world. Also using the knowledge that we have about the DNA genome analysis to help us produce drugs. So if we can't find the genes that code for various useful proteins, like insulin, for example, then we can't genetically modify organisms in order to produce those molecules. So we need that DNA sequencing technology in order to be able to find the genes that we need, in order to be able to extract them and then use them for genetically modifying organisms. New proteins, if we know the sequence of the DNA base sequence, we know then the sequence of the amino acids, we can translate that, and then we can kind of start using this information to actually design and build our own proteins. So it might be a monoclonal antibody, for example, that we need to bind to something specific, and we can manipulate that using this information that we've got. Biosensors. So an example of a biosensor is where we could attach a microorganism to a microchip that can detect changes in the environment and then produce some kind of response. So like a glowing light or something. But to use them, we have to understand how they work, how their genes work, what could trigger the biological changes, or genetically modify them so that they will respond to certain changes in the environment by glowing or producing some kind of signal. So that's really important, and they can be obviously used in various places where we need to make sure we can detect changes in the environment. And finally, nanotechnology. So producing very, 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 very small pieces of technology, materials, substances that are going to work on a nanoscale. This new area of biology has also led to a kind of a new sort of consideration, which is bioethics. So if we know all of this information about organisms and we have all the DNA, especially for different humans, so we have DNA on file, or we have this ability to obviously create and produce new molecules or modify organisms through genetic modification, there needs to be a lot more risks and rewards that need to be managed and controlled and regulated. It's not ethical to create synthetic organisms from scratch, so to build new life using these technology that we have. So we have to kind of regulate that and make sure that no one's abusing these technologies and this newfound ability that we have. So we need to make sure rules are being made and regulations are being made, and that people are controlling where and how this technology is being used.
Ouch! This is why in some videos I explain scratches.